Hi everyone, welcome to week 2 of environmental biology. I want to review what you should have completed last week. So last week you should have completed our start of semester survey, our start of course self-reflection. You should have set up your e-portfolio and posted a link to the class list of links. You have also should have completed the week 1 reading. And finally, you should log the hours that you spent on classwork in your activity log for that week. If you have not completed any of these things, please go back to week 1 and complete them now before continuing on to week 2. This week, our objectives are to gain a better understanding of the scientific process, develop the skills necessary to evaluate a source for credibility, and understand how scientists communicate about their work. What I'm going to be covering in this introductory video is what is a hypothesis in science, what makes a good scientific study, and why peer review is so important. So before we dive in, I want to just briefly touch on what is environmental biology. To me, environmental biology and environmental science is really a dynamic and what we call an interdisciplinary field, which means it is combining aspects of multiple sources of knowledge that integrates both the study of living parts of the environment, so these are organisms, animals, plants, fungi, and non-living parts of the environment, such as the weather or geography, and focuses on solutions to environmental problems. So we are going to be spending a lot of time talking about the interdisciplinarity of environmental biology and on the problems that environmental biology can be applied to. So when I think about environmental biology, I think about a lot of different things. I think about air pollution and air quality. This is Utah. This is where I spent the previous 8 years. And this is where I lived on a good air quality day. And this is where I lived on a poor air quality day. So you can see this is not just something for other places. This is something for us here in the United States. I think about things like wildfires, pollinators, electricity sources, and where we get power sources. And I also think about historical implications of how we've treated people and how that has actually changed the environment around them. So this is a redlining map, and we actually know there's been a number of studies in environmental biology that have demonstrated that people who live in formerly redlined neighborhoods have less access to trees. And we'll be talking about the value of trees as we continue our course. But in order to kind of begin integrating all of these aspects of environmental biology, we first need to understand what the scientific process actually is. Often, the scientific process is first thought about as something that happens on a straight line. You start with an idea, you formulate a hypothesis, you do an experiment, and you end up with a result. But the reality of the scientific process is that it is an ongoing and circular process. Often scientists are making observations and thinking about interesting questions. They then take those interesting questions and they turn them into hypotheses, which is a prediction about what causes a phenomenon I'm observing. After developing that into a testable hypothesis, we gather data in an experiment, and then we analyze it to develop either general theories, or perhaps we evaluate it and we find that we need to go back and adjust or reject our hypothesis and start over. So this is all a circular process, and even after we've analyzed and developed our general theory, that leads us to new observations, which starts the whole process, the whole cycle over again. So when we talk about formulating hypotheses, what we are actually talking about is developing a prediction about the observations that we happen to see. So what is a hypothesis? At its very basic level, a hypothesis is really a prediction about what we will expect to happen. Okay? And when we develop a hypothesis based on findings from previous research. So it's not simply just I have a prediction. Instead, we spend time as scientists making a case for why a hypothesis or a prediction should be made in the first place before even diving into it. And the next really critical part after we've built this hypothesis based on previous findings is that a hypothesis must actually be testable. If it cannot be tested, it is not a scientific hypothesis. And finally, a hypothesis needs to be falsifiable. So you need to both be able to test that hypothesis, and there needs to be a way of proving it wrong. If your hypothesis is that fairies will cause this plant to grow, and so my prediction is that this plant will grow because of the fairies. If there is no way to test if fairies are present because you tell me that they are invisible, then this hypothesis is not testable. If you tell me that my presence will scare away any fairies to prevent that plant from growing, that hypothesis is not falsifiable. So that is one example of a very out there example, hypothetical example of a hypothesis that is not a scientific hypothesis. After we develop our testable, falsifiable hypothesis, we go and we actually conduct experiments. Okay. So this means we gather our data to test predictions and things that make a good experiment. So there are certain qualifications to what we see as a good experiment in science is that it should have a control. It should have either something that is a prior example of what this phenomenon is without the impact that we're testing, or we need to have two different treatments where there is one group that gets the treatment and one group that does not get the treatment. Additionally, experimental variables need to exist within our experiment, and they must be quantifiable, so we need to be able to actually measure those in some way. And finally, one of the most important parts about a good scientific experiment is that it can be repeated or replicated. That means that we have multiple trials to draw our conclusions from, so we know that it is not just a one-off situation that we had, that we were drawing our conclusions from. After we've collected our data, we analyze it and we develop our general theories. We put this into a publication and we share our results. And as scientists, it's really important that we share our results. And we do this in a very controlled way. So these results go to a journal and it goes through a process called peer review. So whenever I have an experiment and I've written it up and completed my experiment, I write this whole thing, I send it to a journal that is appropriate for my experiment. Maybe it's the Journal of Ecology, maybe it's the Journal of Global Change Biology. But the editor there then says, wow, Dr. Weglers, this is a really interesting study and it fits my journal. So I am going to contact a whole bunch of other scientists and ask them if they will review it, who did not write this paper. And so that editor takes my paper, sends it off to my colleagues who did not write this paper, so other scientists, other experts in the field, and they go through it very carefully and make sure that that this study actually qualifies to be a good scientific experiment. From there, they come back with edits. My editor sends this back with the edits that the reviewers want to see, and I edit it appropriately before it even goes to publication. So this whole process helps science ensure that scientific studies that are done are being done at a highly rigorous level. So the scientific process is very circular, so it feeds into itself, so it's always growing and always continuing, and also it has multiple checkpoints in it. After I've published my results, what I have done is I've actually shared my results with the scientific community. So this is one way that we participate in what we call scientific communication. Scientific communication is the communication of scientific findings both to other scientists, so this would be publishing peer-reviewed articles, and also with society as a whole. So we may do this by communicating with scientific journalists, other actors, so this could be people on the internet, or PR and press officers at a university. All of these things and the way we take a scientific experiment and share the results with others, that is considered scientific communication. But scientific communication can go wrong because, as you can see, there are multiple different players and actors within this, this idea of scientific communication. And so sometimes science uses a whole lot of specialized words, and we refer to these as jargon. And sometimes when people review an article without talking to the scientist who did it, what we end up with is an article that does not actually reflect the findings of the report. So this is one really great example that I know of. There was a study that was done on monarch butterflies looking at potential for extinction risk and population targets, and what populations we need to keep our monarch butterflies at in order to avoid extinction. And this was actually taken, and you can see GMO-linked herbicide may doom monarch butterflies. There was barely a mention of a GMO-linked herbicide in this manuscript of this experiment, but a news outlet happened to pick up on that, and it really took it and ran away with it, and it completely misrepresented what this study was about. So scientific communication can definitely go off the rails at times. And we are going to be talking about that in a little bit more depth later on in our Plato page this week. So I'm really excited to start talking about this and start hearing from you, and I hope this has been a great intro to what